Sorry. I may cut out some of this, to be perfectly honest.
Yeah, boy.
I feel bad.
I learned a tautology.
That may be the title, though.
Yeah.
I feel like that might be.
I think it has to be. I think it has to be.
This is Audiomunity, a podcast about our body's never-ending fight with the outside world.
Hi, everyone. Welcome to Audiomunity. This is episode 50. We are here recording on December 4th. My name is Matthew Woodruff, an assistant professor at Emory University, joined today by co-host Kevin Bonham.
Hello, hello.
And today, episode 50 is brought to you by pile drivers. Pile drivers. We drive piles. The reason we're sponsored by pile drivers today is you may hear in the background a faint ringing, repetitive ringing.
Oh, that's good news, because there is half a block. It just stopped. Half a block from my office, there is some construction happening where they're literally taking like 50-foot steel rebar and just pounding it into the ground with some kind of hydraulic machine. It's wild. I got a video of it. I may splice it in to this video, although maybe not for the audio version because that would be horrifying. But yeah. It's been going for weeks, and it sucks. Okay. Shakes the whole building. If you hear that while I'm speaking, I apologize. I promise you it is worse for me than it is for you.
Yeah. I will say that we've already started over this episode because it was too distracting to say that first sentence through the repetitive banging. So let's just assume...
There it goes again.
There we go. Let's just assume that that's going to happen a couple times. You may hear that throughout. I may trail off mid-sentence, forgetting what I'm talking about.
I will do my best to make sure that I am muted during the times that I am not speaking. But yeah, no guarantees on the audio quality of this particular episode.
Yeah. So speaking of trailing off, perhaps absent-mindedly, actually today we are continuing the, I think the trend, maybe, because 2 is not really a trend, but we are going to sort of leave off our last episode where we really flew pretty far afield of immunology and talked about repetitive brain injury and CTE. And we're going to talk about brains a little bit more in a way that we don't generally do. And so we are immunologists, obviously, but I think one of the consistencies through this podcast has been the interesting places that we find immunology where we may not expect it, right? And so we are today going to get into the field of neurodegeneration, which may not seem like it is all that connected to the immune system, except that everything is connected to the immune system. And I think increasingly there is interest within the neurodegeneration community around the role that inflammation plays and the immune system might play in sort of the development and acceleration of disease. And so, this is a space that we haven't really operated much in before. We've talked, I think, about brain biology at some level in past episodes, but never really...
We've talked about lymphatics in the brain. We've talked about... We had that weird paper from a few episodes ago now where we talked about T cells navigating from the gut to the brain and then influencing behavior. We talked about headbutting. And this is neurodegeneration that has nothing to do with slamming your horns together or mountain goats jumping off cliffs and landing horn first in the ground. Has nothing to do with that. But yeah, I think neuroinflammation and the links between the immune system and the brain are quite interesting. I mean, my main area of study now is the gut microbiome in the brain. A lot of that connection is potentially mediated through the immune system. And also, everybody wants interdisciplinary. Field boundaries are just made up. So, why not? We can talk about brains on autoimmunity, I think.
Yeah, we're going to do it. So let's start just by talking, because you just mentioned this is not traumatic brain injury. This is not autoimmune-induced brain injury in a traditional sense, the way that we talk about things like MS, and we have talked about MS in the past. This is really a slow degeneration of the neural connections that make our brain function. So, we're going to be talking about Alzheimer's disease today. And, in talking about Alzheimer's disease, we'll talk about a couple other neurodegenerative diseases. But let's start there because that'll be the focus. And we haven't really introduced this topic before. So Alzheimer's disease, first of all, is extraordinarily common. It is the most common cause of dementia. About 60 or 70% of cases of dementia across the world seem or are supposed to be linked to Alzheimer's disease. And we're talking there about 55 plus million people with current aging population. So this is a pretty substantial group that is affected by this. And many people, I think, are familiar with somebody that has gone through the stages of Alzheimer's disease, but if you haven't, basically what we're talking about here is in aging populations, a general decline in memory, which eventually turns into a loss of language skills, usually. And all of this, I'm sort of generalizing. This is obviously patient to patient, and we'll get into why that is in a second. But eventually, that loss of language, decline in memory, leads to declines generally in executive function, and slowly turns into a lack of ability to participate in basically basic self-care, right? So basic daily function declines. And so, when we're talking about this, we're not talking about just forgetfulness. We're talking about pretty significant societal impacts of this disease, caregiver impacts, and it is the top cause of disability and death in older adults. So pretty wide-sweeping implications as to whether we understand this. And it's been a really complicated topic because studying this disease is really hard, both from a patient perspective and also getting access to brains and getting access to cerebral spinal fluid. And, all of those things are just very hard. That said, there have been some pretty significant updates in this field over the last 30 years, especially with the advances in brain imaging. And so, I think it's really an appropriate time to talk about that. So obviously, we want to talk here about the intersection of Alzheimer's disease and the immune system, right? That's sort of what we're here to do. But before we get there, I'd like to talk a little bit, I think we should talk about mechanisms of Alzheimer's disease, like what is actually going on in this disease, how does that compare to other neurodegenerative disorders, and an important component of this is not just in the brain, it's actually in the vasculature of the brain. So we'll talk a little bit about some of the vascular contributions to cognitive decline that kind of jumps into this picture in most patients. And then finally, and this is where we will get to our paper, we're going to talk about a gene called ApoE4. And ApoE4 is a lipoprotein that has its fingers sort of all over this process, both on the cognitive side and also on the vascular side. So that's sort of where we're headed. But I think the most important place to start is, how does Alzheimer's disease actually develop? Like, what is going on when someone's brain is starting to lose its function? And the answer is proteins are misfolding. It's really as simple as that. Obviously, the picture is a lot more complicated, and as we get into other neurodiseases, we find that it's not always the same proteins. But at the base, proteins are misfolding here. When proteins misfold, cells die or lose function, and that's what we're talking about today.
And it might be worth saying that, like, protein folding and misfolding is all over the place in our bodies. This is just like a constant thing. Like, the fact that these long strings of amino acids fold up into correct shapes is actually pretty remarkable, like just in general. And all of our cells depend on this. Proteins are constantly being misfolded to some greater or lesser degree. When there are cellular mechanisms to try to refold misfolded proteins, there's cellular mechanisms to degrade misfolded proteins and make sure they don't clog stuff up. But the easiest thing to do if protein unfolding or misfolding gets out of control is just to have that cell die and be replaced by another cell. The reason this gets really important in the brain specifically is because, of course, neurons should be very long-lived. You cannot simply replace a neuron and all of its axonal and dendritic connections to all of its neighboring cells, right? So the connections between neurons really matters in the brain. And so it's not quite so easy as just killing the cell and replacing it with, like, if you're a macrophage and you gobble up a bunch of misfolded proteins and you're kind of a little, like, yeah, macrophages are long-lived, but it's okay if that macrophage dies and gets replaced by a new macrophage, for the most part. In the brain, that's much less true. It's true to some degree. Like, neurons are dying all the time. Neurons are getting replaced, but the fact of the structure and connections between neurons being such an important component of their function means that it's not that protein folding is not a huge problem everywhere else in the body. It's just that it's much easier to deal with in other parts of the body than it is in the brain.
Right. And so when we're talking about protein folding within the context of Alzheimer's disease, we're really talking about two different proteins. And people may have heard certainly of the first because it's been the topic of a lot of conversation. This is, of course, amyloid beta, right? So amyloid beta is the protein that all of the new Alzheimer's medications that went through FDA clearance, all the antibodies that targeted Alzheimer's, those antibodies, that drug targets amyloid beta plaques, right? And so this is a small peptide that is derived from a larger protein, APP. This is, as Kevin just said, this is a normal protein. It exists in brains. It actually is important in brain repair. But if it builds up, if you get an accumulation of this small peptide called amyloid beta, it oligomerizes, right? So you go from one amyloid beta peptide to two combined, and then it just gets bigger and bigger and bigger, and it forms these much larger aggregate structures that sort of deposits. Now, we just talked about lymphatics in the brain. That's something we've talked about before. There's a couple mechanisms where you would normally expect that this stuff would get flushed out, right? Lymphatic flow through the brain would be one of those expectations. Microglia, the macrophages of the brain eating these things, is another. But basically, in Alzheimer's disease, we see the accumulation of these large oligomeric structures of amyloid beta deposited directly in the brain in a way that you can actually visualize them through PET scans.
I just want to say, like, that idea of protein sort of misfolding and sort of like starting to oligomerize, it's quite analogous to other things people might have heard of as just prion disease, where you have these sort of foreign proteins that are misfolded and they sort of aggregate other misfoldings together. And it's no surprise that, like, mad cow disease, as it's commonly known, for some prion diseases, there's the one that's associated with cannibalism. I can't remember what that one's called. That's on the tip of my tongue, but it doesn't. But it's no surprise that those diseases also show up as brain misfunction. Again, it's not that those prions are not going to other parts of the body and causing the same problems. It's just that it's much easier to deal with elsewhere than it is in the brain.
I have good news though. Prion diseases have been fixed, and I know that because there's no more division studying them at the CDC.
Oh, good.
Yeah, so we got that under control.
Just as you said that, the pile driver started.
That's exactly right. So that's one, right? Amyloid beta is one. The second protein that we talk about is tau. And tau actually is not a peptide associated with the outside of the cell. Now we're talking about proteins that are inside the neurons themselves. And anybody that's seen a picture of the neurons knows that they are these long, delicate structures, right? They have lots and lots of different connections. As you can imagine, there's a lot of internal infrastructure that needs to keep the cell stable in that long, delicate shape and maintain all of that. One of the proteins that is responsible for that is a protein called tau. Tau is an infrastructure protein. We have microtubules that sort of elongate those cells and keep everything in the right shape. Tau basically helps stabilize those microtubules in that state. Right?
Just pause for a digression. Microtubules, kind of like giant steel beams. Thankfully, in neurons, we do not require hydraulics to smash them into anything to stabilize them.
No, it's a good point. Although perhaps we should.
Perhaps
maybe
we should. Tau
window.
is not a pile driver.
Good.
Tau. We did discuss tau in the last episode, talking about how phosphorylated tau is sort of associated with neurodegeneration.
Yeah, and so, and you can see it, right? We talked about being able to see this stuff. You can actually see tangles of tau in these cells. They call them neurofibrillary tangles. So again, visualizable, hard to see without a brain dissection, right? And this is, again, part of the reason why it's so hard to study this stuff is you actually need brains generally in order to get into the pathology here. So those are the
and
two
Nicole
main
was saying
proteins.
that you can't actually see it macroscopically or even under a microscope, I guess, until it's gotten to the point where it's so bad that you're already seeing dysfunction.
Yeah, you're talking about one intracellular tangle, right? Like that's
right.
and obviously that's propagating out. So those are the two proteins that we're talking about. But in addition to that, when we're talking about Alzheimer's disease mechanism, right, we're also talking about, of course, these bystander microglial cells, right, which are trying to respond to this environment. They're seeing the tangles, they're seeing the plaques, they're trying to keep the area clean. That's their job. And we know over and over again from other episodes on autoimmunity that if macrophages are trying to clean things that they cannot clean, they get angry about that. And the result of that is localized inflammation, right? So we have a less efficient amyloid clearance mechanism, increased cytokines, reactive oxygen species, which eventually ultimately leads in synaptic pruning in the surrounding neurons, right? And if you look at the Alzheimer's disease risk genes, it's things like CD33, TREM2, ApoE, and I know that those things don't mean anything to the folks that we're talking to, but these are genes that are associated with microglia and macrophages in general, right? So this there are predispositions that show up in the immune system in the brain related to the ability to effectively deal with these plaques without causing a ton of inflammation, and of course, chronic inflammation will amplify any of this, right? Risk factors that result in systemic inflammation, obesity, smoking, these kinds of things all sort of manifest in this same way. And ultimately, we've talked about it already, but all of these three things together, right? The accumulation of amyloid beta plaques, tau, tangles, chronic inflammation in the brain is what ultimately leads to this synaptic pruning and loss of connection between basically areas of the brain, between individual neurons, which leads to memory loss, right? Language disruption, everything that we talked about before. So how does that compare to other mechanisms of neurodegeneration, other neurodegenerative diseases? And the answer is that, this is not unique. Alzheimer's disease is not unique when we're talking about protein misfolding and accumulation. But basically, the thing that discriminates Alzheimer's from other diseases is what proteins are being misfolded and where are those proteins misfolding in the brain, right? So another good place to talk about neurodegeneration is Parkinson's disease, which is another pretty widespread neurodegenerative disorder. But when we're talking about Parkinson's disease, we're not talking about amyloid beta anymore. Now we're talking about a protein called alpha-synuclein, right? Again, we're talking misfolded proteins, but we're talking about depositing those misfolded proteins instead of in the cortical region, right, in the brainstem and basal ganglia, which means that motor control is going to be the first thing that goes, right? So it's not memory impairment because it's not the part of the brain that does memory. It's motor control because you're getting deposition in the part of your brain that coordinates your motor function, right? And so this is true across a number of different neurodegenerative diseases. Frontotemporal dementia is another disease that folks talk about. We're talking about some tau involvement, but also a protein TDP43. And there you're looking at behavioral changes as the first thing that happens, along with progressive aphasia, right? And so again, which proteins are being tangled, which proteins are misfolding, and where are those proteins in the brain? So getting back to Alzheimer's disease, the thing that makes Alzheimer's disease and not Parkinson's disease is that the dominant feature is memory impairment because of where it deposits in the brain, and it is this combined amyloid beta and tau pathology that dominates the disease. Now, an important component of this is we're going to talk about these things as if they are all separate from each other, and frequently they're not. Frequently, you can have complicated versions of these things that all have different components of these diseases, and part of the reason for that is that the fundamental mechanisms are frequently the same, right? We talked about vascular involvement, which we'll talk about a little bit more, right? But vascular involvement is systemic in the brain. It can hit multiple locations, right? So this picture of neurodegeneration can actually get quite complicated, and you'll get conflicting diagnoses or multiple diagnoses, right? It is not impossible to have multiple things going on.
Yeah, and I think another piece of that, sort of the converse of that idea that all of these different dementias or brain pathologies are related, is that the things that we lump under Alzheimer's disease, there may be multiple things, like multiple different causes that lead to the same outcome that then gets labeled as Alzheimer's disease. But if you are, whatever process is leading to the accumulation of beta amyloid plaques and tau in memory centers in the brain, that's going to get labeled as Alzheimer's disease. But we don't actually know formally that they all have sort of the same cause. It's kind of like autoimmunity. Like there's ways in which certain kinds of predispositions will lead to autoimmunity that hits the brain versus causes, lupus and antibody pathologies or inflammatory arthritis. There are sort of trends that will lead to a particular manifestation, but there may be multiple different causes that all kind of end up in a similar pathology. And that's one of the things that makes these things so hard to study. We don't know what the instigating cause is. What we see is something that's already far downstream of the initial insult that sort of ended up compounding and leading to the symptoms that we see. So I think it's worth noting that in this paper, we're going to talk about one specific potential instigating cause that seems to be associated with a lot of different kinds of pathologies, but it can be the case that you have multiple causes that lead to a similar disease, or an initial cause that, because of somebody's other physiology, will lead to other pathologies instead or at the same time.
Yeah, and I would add to that. I think I agree with everything you said. One of the things that makes Alzheimer's disease unique is that strong genetic drivers have actually been identified for this set of symptoms, right? And one of the reasons why we want to touch on ApoE4 is because ApoE4, which is, again, the gene that we're going to talk about today as a potential causative agent here, is highly prevalent in the human population and also is seemingly causative in a way that very few other neurodegenerative diseases have, right? So you'll have like one-off genetic mutation events, certainly, that show up in populations, but this is one where there's a really high penetrance, and that makes Alzheimer's disease somewhat unique and also interestingly studyable, right? Because it means that you can identify folks that have ApoE4, and we'll talk about this in a minute, have some sense of what the trajectory is going to look like and hopefully intervene.
And actually, I want to just have a quick clarification terminology point. So the gene and the protein are Apolipoprotein E. There's a couple of different apolipoproteins. ApoB is also commonly referred to. When we say ApoE4, we are talking about a particular variant of the gene, right? Everybody, all humans, have ApoE genes, 2 copies.
Yes.
There is a variant called epsilon 4 that is the allele, the version of ApoE that is associated with disease. And so we're going to say ApoE4 a bunch, and I wanted to clarify that it's not that these people have a gene or a protein that nobody else has. It's just that it's a variant of a gene that everybody has.
Right. And I mentioned that, ApoE4 is certainly going to be related to the development of these protein plaques, right, that build misfolded protein plaques, but also that there is a component of vascular process here that must also be talked about when we're talking about neurodegeneration, right? So ApoE4 is not only a risk factor for the accumulation of protein aggregates, but it is also a risk factor for vascular processes, right, that also contribute to dementia. So we know that in dementia, in neurodegeneration, there's a significant component of small vessel disease where we get microbleeds in the brain, right, at various locations. There are extreme versions of that. Things like cerebral amyloid angiopathy, or CAA, where we actually have amyloid deposition directly in the vessel walls, right? Because ApoE4, we're going to talk about it in the brain, but this is a systemic protein, right? It's attached to lipid droplets being shuttled around your body at all times, right? So we actually get amyloid beta deposition in the vessel walls, which frequently overlaps with Alzheimer's disease. So these two things are not separate from each other. You can have Alzheimer's disease with or without CAA. And if you have CAA as well, that's an accelerating factor. And generally speaking, and we've talked about this a lot, overall, what you're looking at is a breakdown of the blood-brain barrier, which means that your brain is now exposed to a lot of the systemic inflammation events that we talk about all the time on this show, right? So whereas somebody with a healthy blood-brain barrier might, have a fever or an infection or something like that that's mostly compartmentalized outside of the brain, perhaps those cytokines find a way to get in. So we're just talking about the ability for the system, the immune system, to start to impact the brain in ways that are going to eventually be detrimental. Of course, I mentioned before, because this is vascular disease that we're talking about, all of our other overlapping risk factors that we associate with vascular disease will also contribute to these processes: hypertension, smoking, hyperlipidemia, all of these things. And so, one of the major discussions around neurodegeneration is if you are predisposed, right, mitigating those other factors that actually impact the vascular side very heavily, right? Because, you may or may not be able to control deposition in the brain, but with a healthy vascular system, you can actually mitigate a lot of the risks associated with at least the blood component of this. Although, we talk about the lymphatics in the brain a lot. And one of the things that we talk about is its use as sort of a clearance mechanism. And so healthy blood flow to the brain, right, through these systems will actually at some level almost certainly contribute to lymphatic flow. And so, completely disconnecting these two things, right, the accumulation of proteins in the brain and the health of the blood vessels is really impossible because the two things are intimately connected.
Yeah, and similar to that link with vascular disease, I think metabolic disease is also has a lot of similar systemic effects, right? So we're talking about lipoproteins here. These are as Matt mentioned, there are these sort of people are probably familiar with on their blood tests, like HDL and LDL, these different sort of cholesterol-containing things that shuttle fats around from your liver. If you have metabolic disease, things like diabetes or prediabetes, insulin insensitivity, and again, there's like genetic risk factors, there's behavioral risk factors for these things as well. All of those things can also contribute to a different metabolic balance that will affect the composition of your blood in terms of metabolic products that are moving around, and can also have systemic effects, including inflammation. So like these things are broadly connected. It is very hard to isolate individual causes from individual effects. But one thing that we see is that certain variants of these apolipoprotein genes seem to be associated with a bunch of different potential outcomes that are sort of connected to all of these different body systems.
Exactly. And so, we've touched on a couple of those sort of interlocking systems that show up in neurodegeneration. And the reason that we're interested in ApoE4 is because it seems to touch all of those systems, right? We said that it's a rare, true genetic driver, it seems, of neurodegeneration. And it seems to be because it hits proteinopathy, right? It promotes amyloid beta deposition. It reduces amyloid beta clearance. We know that from mouse modeling. In neuroinflammation, it seems to reprogram microglia towards a more inflammatory and lipid burdened state, right? So a little less immune quiet, a little more inflammatory. And it alters the response to brain injury and plaque development within those microglia. And then finally, as we just talked about, vascular function. We know that ApoE4 is a risk factor for declining blood-brain barrier integrity. It increases vulnerability to small vessel disease and microbleeds in the brain, and it worsens outcomes after stroke. So all three of those components that we just talked about as sort of contributing factors to neurodegeneration are all touched by this ApoE4 allele, which is why it makes it so interesting. And it also makes it more studyable if you were able to identify a true disease, a true classification, identify those patients, and then do longitudinal studies, right? It sort of makes it unique within this neurodegeneration space. And that brings us to 2024. So everything that we have talked about so far is, decades worth of work, identifying Alzheimer's disease, classifying it, talking about pathology, identifying ApoE4 as a risk factor, all of those things. But it actually wasn't until 2024 that a paper came out saying, look, there are unique factors here within ApoE4 individuals where we think that we should truly classify ApoE4-driven Alzheimer's disease as its own compartment of neurodegenerative disorders, right? So that we can identify those patients, start classifying them, and do directed studies. There was a seminal paper that came out in 2024 called ApoE4 Homozygosity Represents a Distinct Genetic Form of Alzheimer's Disease. And I'm not going to go into that paper in any great detail, but a couple things that they showed in that. First, that ApoE4 homozygosity has an extremely high penetrance of disease. You're talking about basically 100% of people by age 65 showing some indications of biological effect of having that ApoE4 allele. So biomarkers, for example, in the blood or the cerebral spinal fluid. There is a consistent and pretty predictable age of symptom onset. So most people are experiencing mild cognitive impairment around 65 years old. There is a very predictable sequence of biomarker changes, which makes it extraordinarily useful for drug and clinical intervention studies, right? So you start to see the accumulation of biomarkers somewhere around age 55. And so that's a full decade before what I just mentioned, which is symptom onset at about age 65 with that mild cognitive impairment. We know that there is a phenotype here that more resembles the quote-unquote genetic Alzheimer's, however we want to classify that, than the sort of typical sporadic Alzheimer's disease that shows up in the population. So that sort of made it unique. And I would say that the take-home from all of that is that there was strong evidence in that paper that ApoE4 is more than a risk modifier. It is causal in most cases. And it sort of challenged the view of ApoE4 as a, one component of a larger system. It does seem like for homozygous patients, it is a driver and can act through those different pathways that we talked about and leads to a relatively stable and repeatable phenotype. Now, again, everything that you just said, Kevin, doesn't stop being true, because this is still a multifactorial disease. It's just that this one factor seems to be able to drive at least this set of symptoms. And that does not mean that there are other drivers, there are not modifying factors, there are not environmental factors. It just means that this has a particularly potent effect on the generation of neurodegeneration. So that is a longer than average intro than we do here at Autoimmunity, and the reason for that is I do think that neurodegeneration is complicated. It is its own field before we start talking about the immune system, but of course it is Autoimmunity. We are interested in the immune components, and so the paper that we've decided to talk about today is since 2024, right? Since we have decided that ApoE4-driven Alzheimer's disease is kind of its own thing, there's a lot of interest around what exactly that means. And particularly, we are interested in the neuroinflammation side here. So the paper we are talking about today is ApoE4 carriers share immune-related proteomic changes across neurodegenerative diseases. So this is a look at ApoE4, the allele, like Kevin was just talking about, and the signatures associated with ApoE4-driven inflammation. First author of the paper is Artur Shvedkov, and I hope that I got that right. I believe that I did. And the last author here is Caitlin Finney.
And they're both co-corresponding authors, first and last.
They are indeed. And so, yeah, this is a paper that was published in Nature Medicine. It's relatively recent, August of 2025. So, I mean, we're considering this a really high impact work, and I think that the reason for that is pretty clear to me. Just looking at study design here, we've talked about how hard it is to track patients, to gather patients, to study patients. It is really valuable to have stable patient sets to work on. And the result of that is there are large data banks, biobanks across, not just, obviously neurodegeneration, but a lot of different disease states now where researchers have sort of pulled together resources across the country and across the world and tried to share those resources as best they can in order to get real answers. And so what we're looking at here is a huge cohort, right? A huge cohort of neurodegenerative diseases. I think that their initial cohort looks like 11,270 patient samples or patients. Yeah,
and this is not terribly uncommon for like genetic studies. Which is sort of what this is derived from. So lots of like big genetics. If you hear about like genome-wide association studies, for example, these have to be enormous cohorts, basically in order to get statistical associations that are meaningful. You often have these cohorts that are tens or even hundreds of thousands of people involved. I think the thing that's kind of amazing about this is the combination of the genetics, which is like, it's not easy to do 100,000 people genetics, but it's not as hard as also getting biosamples for very large numbers of these people. So we have the genetics, and then also the associations, which you need for doing normal sort of GWAS things. But then in addition, you've got these huge numbers of biosamples associated with these subjects where they're going to look at proteins. To me, that's the thing that's so remarkable about this paper.
Plasma is one thing, right? Pulling blood is one thing. This paper starts off by doing a soma scan. So this is a protein scan of about 6,500 proteins. So basically doing not full proteomics, but pretty good depth proteomics on cerebral spinal fluid samples, right? So you're talking about tapping 11,000 people. And that is not a non-invasive procedure. It's not a great procedure to put folks through. It's really important.
Well, the CSF is only for less than 1,000 of them.
Well, that's a good point.
So they're looking specifically in the CSF. It is more than 1,000. So it's 526 people with Alzheimer's disease, 247 with Parkinson's disease, and then 573 what they're calling non-impaired control individuals. So it's more than 1,000.
Which is a lot.
Which is still, it's a lot. That's a lot of people to get CSF from. It's not 11,000, but it's still a lot.
It's a lot.
Yeah.
And so
I've never been on a paper with a study that had more than 1,000 people on it.
No.
And I've done a bunch of these sort of epidemiological kinds of things.
Yeah. No, it's a large effort. It's a large effort. So then they did that, right? So they took those proteins, they took that CSF for that 1,000 people, and they ran this soma scan, and they thought to themselves, here we go. And then they got the PCA from figure 1b.
Yeah.
So figure 1b breaks it down into ApoE for positive Alzheimer's disease patients, Parkinson's disease patients, and non-impaired patients. And then obviously ApoE for negative versions of those three things. And for those of you that aren't looking at figure 1b, what you see here is a non-stratified cloud of all of those patients, which are clearly not discernible through the
technical definition here is big fucking blob.
Yes, they got a blob, which did not appear to show any real organization based on the proteomics of the CSF.
And I want to be fair here. It is not necessarily the case that you would expect a huge variation on a PCA like this. With this many proteins, most of those proteins are not going to be directly involved. And so, this isn't terribly surprising. I would have been surprised if you saw a clear separation here.
Okay.
So, just
I would have, as a grad student, seeing this PCA for the first time, having just run 1,000 of these submitted, which I'm sure was ungodly costly, I would have been sad to see figure 1b. There's no doubt in my mind.
This would not have been figure 1b for me, personally. Yes.
Yeah. So then they say, well, that's, not what we hoped, but we can do more than that, right? We can dig a little deeper. And they do some statistical magic, which is functionally correlation testing. But I'm gonna have Kevin talk about this because this is where Matt's expertise no longer matters.
Yeah, so they use a measure called mutual information, and I was trying to think of a good analogy for this. So mutual information is akin to correlation, but it allows you to capture nonlinear relationships. So the analogy I was thinking about, mutual information, the idea is if you know something about one variable, how much does that tell you about another variable? So the analogy I thought of is temperature. So imagine that you have a big data set of all kinds of weather-related variables. And you ask, what is related to temperature? So if I measure the temperature in Celsius, the mutual information with temperature in Fahrenheit is 1. It is if I know the temperature in Celsius, I know everything that I need to know about the temperature in Fahrenheit, right? It's a direct connection.
Yeah.
Everything about temperature in Fahrenheit is contained in temperature in Celsius. If I ask you, what is the temperature in Celsius tell me about the phase of the moon? It tells me nothing, right? Phase of the moon is unconnected. It has a different cycle from day-night. It has a different cycle from seasonality, etc. So I have no relationship between temperature and phase of the moon. Now, if I ask you, if I know the temperature, how much information do I have about time of day that the temperature was measured? I have some information, right? It is not a linear relationship. So if I'm measuring time from in 24-hour cycle, 0 to 24.9 or whatever, 0, the bottom of that scale, up through let's say 5 hours, is kind of similar in terms of what it tells me about temperature as 20 to 24, that's the nighttime. Nighttime tends to be colder. So there's not a linear relationship between time of day and temperature, but knowing the temperature does give me some information about the time of day that it was measured, right? If I measure a temperature of 112 degrees Fahrenheit, probably not going to be midnight in general, right? So mutual information is just a way of calculating relationships between variables that are not necessarily linearly correlated or even rank correlated. Like if you do a Spearman correlation, for example, it doesn't have to be a linear relationship. It has to be what's called monotonic, where it sort of has a similar directionality. Mutual information is a way to calculate the amount of variance from one variable that is associated with variance in the other variable. So, got it. Maybe more information than we need to, but I think that's sort of the foundation of this entire paper. So I think it's worth saying it is a useful measure for things that might be non-linearly correlated.
Yeah. No, that all makes sense.
It is still a measure of relationships between things.
No, I think that makes a lot of sense. And I think you're right. It sort of forms the basis of the whole rest of the paper. So that was a good analogy. I applaud you for the temperature analogy. That was good.
Thank you.
So great. So now what we can do with that information in hand is we can ask the question, does ApoE4 or do any of these disease states tell us anything about these genes? The expression of the presence of these proteins in the CSF. And of course, the answer to that is yes. They pulled out 229 proteins where they say the disease state or the disease classification scheme here does tell us something about how these proteins are behaving. So they set a threshold, they set a cutoff, and they say here are the 229 genes most correlated, either linearly or not, with the presence of disease, and then they ran the same PCA again. And this time...
By magic, this is amazing, correlated things are correlated.
I don't...
Wow. Sorry.
That was glib.
It's me.
That was quite
glib.
It's
mean, but...
Yeah. Yeah, no, I mean that's literally what that's what this is, right? So what we did was we picked the 229 genes that are most differentially expressed or most correlated. They're not differentially expressed. They're most correlated with the features, and then we see that the features segregate based on those genes, right?
Yeah.
And what we see, and I think that maybe this was the interesting bit, it's that they didn't segregate into disease states necessarily. They actually segregated into ApoE4 presence or not. And I think that probably was the part that was most surprising.
It's not surprising because the way that they identified ApoE-associated proteins was based on the genetic whether the allele was there, right? ApoE associated means that information about these proteins gives you information about ApoE status. Then they cluster by ApoE status. Like, that's like by construction. Like, it could not be other than this.
Right.
To be clear. And there's, I almost did this. I didn't quite go this far because I was trying to give these authors the benefit of the doubt, but like, I am pretty sure, I may do this before I post the episode. If I generated 10,000 random vectors, then I calculated mutual information, picked the top 200, and then did a PCA, I'm pretty sure that I would get this result. Because, like, if you just randomly generate numbers, like, yes, in principle, on average, mutual information between randomly generated numbers will be 0. But just by random chance, when you do this, some of your vectors are going to tend to be more associated with other random vectors if you have enough random vectors, right? So it's not that you will never find any mutual information. It's that you're going to have some distribution that's going to cluster around 0. But if you set the cutoff, as they did, at 0.01 for mutual information, right? This is a scale that goes from 0 to 1. Their cutoff was 0.01. There's no p-value here, so you can't do things like multiple hypothesis correction or things like that you would do if you were doing something that generates a p-value. So there's no correction for the fact that they're looking at thousands of variables. And they find a pretty small number out of their more than 6,000 proteins that are associated. 229, and in later parts it's even less than that. But then if you do that and you find the things that are associated, and then you cluster, or you do a PCA, you will by definition have the first axis be along the thing that you were looking at these things because they have mutual information. Like, it just is that way. It has to be.
So we're not confident then that C or D really tells us much outside of the fact that you found 229
they can do math.
Associations, right? So I guess maybe that's the takeaway, right? Is that does ApoE4 status tell you anything about protein content in the cerebral spinal fluid? And the answer seems to be yes. You can find proteins that are correlated with that genotype. However, perhaps unsurprising that when you then use those 229 proteins to cluster by disease state, it's not really going to tell you much more outside.
Yeah, and if I'm being totally fair, they do the mutual information on their unimpaired controls, and then these PCAs are with the entire cohort that includes AD and PD subjects. So if I want to be the most generous, what I can say is that the protein associations that you see in non-impaired controls seem to carry forward into the folks that are manifesting disease. So that is, that's kind of useful information. The problem is that it kind of undermines the idea that there's some signature in the proteins that is going to tell you something unique about the disease status, right? Because if the non-impaired controls that have the same ApoE genotype look the same as the folks with AD or PD that have the same genotype, then that says that the proteins that you're measuring don't actually matter. in some sense to their disease diagnosis, right?
Yeah, or they matter in a stable and chronic way, and not in an acute way that varies with actual disease state,
of
right?
course.
So, what you're
you're not showing any relationship between those,
right?
Not that it doesn't exist, but that you're not showing it.
Yeah, you're talking about environmental components rather than acute disease components, right? I think that's fair. And so we then go into another measure functionally of correlation, which we don't have to talk about in E because, I mean, we're talking about exactly the same thing, right? The things that we have already identified as correlated continue to be correlated in a heat map as well. We have some, assessment of associations. We don't have to get into that either, but I think I kind of
think we have to get into F.
Yeah, I actually okay. Go ahead.
Okay, so in F they're doing a kind of machine learning. It's a tree-based classifier. It's called CART. Doesn't matter. It's not random forest, but it's like similar to that. So what you see, if you're not looking at the paper, what you see is a plot for Alzheimer's disease, Parkinson's disease, and sex, which are variables that they looked at to see whether, if you stratify by these variables, can you train a machine learning classifier to predict ApoE status based on these 229 proteins. So again, we're saying we have feature selected on the whole cohort, or at least on the healthy cohort. But we've seen in the PCAs that those are related, right? It's the same across these. So you've done feature selection on a group of features based on their association with a variable, and then you're saying I'm training a machine learning classifier to predict genotype based on these things that I know are associated with the genotype. And anytime they plot both the train and test set performance, which are all above 0.9, if you ever do machine learning and your training set has the same performance as your test set, you are doing something wrong. I mean, in principle, it means you have a perfect model that is not at all overfit. It is exactly what it's supposed to be. I have never seen that in my entire career of doing machine learning. Training set performance and test set performance should not be that precise and that exactly the same.
I mean,
it just shouldn't happen.
Yeah. What if you had a couple genes though that truly were on off, right? Like if you, let's say, in one of those proteins is an Alzheimer's protein, right? And you either have the protein or you do not have the protein, right? In that case,
I don't need machine learning to tell me that the temperature in Celsius is the same as the temperature in Fahrenheit.
Yeah. So perhaps we didn't need the model here. Is what you're suggesting?
Yeah. I'm saying that this model, if it were telling you something new, it would not have this performance. That's what I'm saying.
I see.
If I find a bunch of correlated things and I throw them into a thing and ask, can I predict the thing all these things are correlated with? It's like, obviously yes.
Right.
You've
already done that work.
Exactly.
By identifying the mutual information, you have done the work that this might have told you. I understand. And so I do think, where this becomes useful is in G and H. And not honestly, all that specifically useful, but generally useful, right? And we've done this before in our own work, and I think that it's important to do, right? When you have identified those 229 proteins, there's a couple ways that you can sort of take to a next step, right? One way is by going into that list one by one, as we all do, by the way, one by one, and ask ourselves internally, do I know that protein? Is that protein associated with something that I am biologically interested with? I would say that my internal list of proteins now is probably, I don't know, 700, 1,000 genes maybe out of a 20,000 genome, 20,000 protein set in humans. I'm probably familiar enough with 700 to 1,000 to be like, yeah, I kind of know what that does. And then you see a couple of them together and you're like, oh yeah, I think, but there's a better way to do that. And it's called a GSEA analysis, right? So basically what you're asking is, in these 229 proteins, are they random proteins across the genome, or are these proteins associated with each other in some way that biologists and immunologists have previously identified, right? And so what we're talking about is a pathway analysis. Is a specific biological pathway of proteins more emphasized in this 229 protein set than it probably should be if all of those proteins were just selected randomly from the genome? And the answer is sure. Yeah, there are pathways that show up. And the reason that this paper is in front of us as autoimmunity is because the number one process associated with this when they went for a biological process assessment was a viral process, right? Now, what does that mean? That does not mean that they have detected a virus in this system. It does not mean that these people are virally infected. It means that the proteins that they saw associated in the CSF of these patients are proteins where some other researcher, some other time, looked at a viral infection and ongoing viral response and saw similar proteins to that being expressed. Okay, so that there is no indication of active viral infection here. I want to make that really clear, because if you're coming at this kind of thing for the first time, that might be confusing. Especially confusing because then what they do is they dig a little deeper into the pathway analysis and they ask the question, specifically, are there immunologic processes that we can pull out of this, right? And what they find is that the pathways that they've selected using mutual information, the proteins that have been selected through mutual information, are hepatitis B associated proteins, Kaposi sarcoma, so herpes virus infection, hepatitis C, measles, EBV, before you even get to an actual immune-focused component, which is a TLR signaling pathway component. So that's, danger sensing, and we've talked a lot about that on autoimmunity. So the first 5 hits are all proteins that have been previously associated with active viral responses. Okay? Again, does not mean that these patients have hepatitis B, measles, Kaposi sarcoma, any of those things. It means
it's how they're annotated.
Yeah, it means that the proteins that they're seeing associate or correlated in some way with this ApoE homozygous phenotype are most closely associated with antiviral processes in other
So we're basically, I think, for all of these, we're basically talking about type 1 interferon signaling, right? Or interferon gamma, maybe.
Yes. Yeah. I mean, there's some stuff when we get into the peripheral blood that seems to be a little bit more specific than that. There seems to be a Th17 type response, which is associated with MS and other things that we've talked about. But I mean, B cell receptor signaling pathway is on this list. There's some chemokine signaling pathways, but yeah, my guess is we're talking largely a type 1 interferon type driver, which is a traditional classic antiviral response.
Yeah. So trying to figure out a way to say this that isn't like unduly snarky,
obnoxious.
But yeah, I don't want to be obnoxious.
Good luck.
But what I want to know, so I dug into the methods this morning. I read this paper 2 weeks ago. A bunch of the notations on my PDF were like, WTF? what is happening here? And so this morning I went through again, and I really wanted to like give the authors the benefit of the doubt. So I looked through their methods section pretty closely, and I didn't go try to run their code. I didn't look at this particular R package that they're using to do this pathway enrichment analysis. But in the methods that they're using for this analysis, so let me back up and say like what this test in general, this feature set enrichment analysis, what you're doing is you have some underlying distribution of whatever it is. So in this case, it's protein levels. You have some underlying thing for all proteins, and then you ask for a subset of proteins, does the association with your phenotype of interest tend to fall on one direction or another, right? Like, is it further towards one end of the distribution or to the other end of the distribution amongst all possible proteins? What I was not able to identify based on their methods in the paper, and maybe there's more in the R package or in if they have supplementary methods, but what are we comparing? What distribution are we comparing these pathways to? I think it's just other proteins in the CSF, right? So my question would be, if I asked you to generate a list of top mutual information proteins for any other phenotype, sex, or I ask you to look at, I don't know, what other thing are you routinely measuring? Age, etc. So if I give you some other variable that should not be associated necessarily with any of these proteins, what would be the list of top hits? Given the results earlier that we talked about, that we were critiquing earlier, I am really worried that when you do these big data things, it is very easy for some early minor spurious correlation to sort of propagate, and then you assign much more value to it than you need to. So an example, when we look at genes in the microbiome, we do these big data analysis. The things that often rise to the top are things like tRNA synthesis pathways, central glucose metabolism, and like just things that are like housekeeping genes, essentially. I really want to know if basically, I think everything in this paper needs to be thrown up against a random permutation and do all the same exact analyses and see if anything comes out differently. Because I'm not 100% certain that it would. I am not confident. I mean, even with these, like, the log FDR p-value here, they're so high that I am skeptical that it's really an unbiased measure, right? P-value assumes independence from the initial distribution. I am not convinced of that in this particular case.
Well, I am going to congratulate the authors on not having drawn Kevin as a reviewer for this paper.
Through the magic of podcast recording, we are here, Matt and I, on December 5th, day after. I'm not exactly sure where I'm going to plop this in, but when Matt and I were discussing this paper yesterday, the thing that you are listening to right now, we were pretty harsh and snarky. If you haven't gotten to the harsh and snarkiness, it's coming. If you have already, this is
we may even want to preempt. So
exactly.
I would say that sometimes we get into these papers and it's fun to discuss, and we try to modulate in conversation as best we can, and then every once in a while you sort of get to the end of it, and we always prepare for these podcasts by reading papers, and we read those papers independently. And oftentimes we intentionally don't talk to each other about the paper all that much because, as we're talking on the podcast, it feels better to sort of be approaching it for the first time. So we try to do our homework, we come together, we will sometimes shoot questions back and forth. But oftentimes, as we're doing it and talking to each other, we're gaining information, right? And I think that that's how science works, right?
And we're either like, sometimes we sort of like undermine our previous conclusions because other people drew other conclusions, and sometimes we like amp each other up in terms of our criticism or our praise, right? So.
Yes. Yep. No, all that happens. And so, in a vanishingly small percentage of the time, 1 out of 50 so far, we have felt the need to go back or forward, if you will, and clarify exactly where some of that discussion is coming from. And so Kevin did an amazing job of actually doing some of the things that we talked about yesterday. There are a lot of informatic-based analyses in this paper. In fact, the entire paper is informatics-based analyses. We have or will speak about the generation of data to see, how this kind of analytic approach could actually apply to real data or not apply to real data in some cases. So, Kevin, you did the informatics magic here, so I'll let you just jump in.
Yeah. So basically what I did was I went into table 1, supplementary table 1 of the paper, and I pulled the demographic information. And then I generated a bunch of random data that sort of matched the parameters of the data set that these people collected, but is entirely synthetic and nothing has any relationship to one another.
So we're talking 6,500 proteins in
almost
the
7,000,
patient distributions, almost 7,000 in the patient distributions, and genomic or genetic percentages that are frequencies that are in the set. And for those almost 7,000 proteins, you have just created random data over a normal distribution.
Yeah, so I just used a random number generator to draw from a normal distribution for protein expression, quotes, and made that data. And so we have, or we will talk about the PCA analysis. And we said if you select based on mutual information, that if you pick the top features based on mutual information, by definition, you're going to have a PCA that shows the clustering that they show in the paper. And when I generated literally random data, that didn't happen because when you select the top 50 on mutual information, those top 50, they don't actually have much more mutual information than the rest of the set. And so I think maybe if we picked the top like 3, it might do a split. Actually, I should go back and actually do that. But basically, if you have enough proteins and there is literally no information contained in the protein expression data vis-a-vis ApoE4, then you're not going to see this. But the other thing that I did is I added a little bit of directional noise based on ApoE4 status, in quotes again. This is all synthetic. And when you do that, when there's any biological signal, in quotes again, at all, if there's any, well, I shouldn't even say biological signal, if there's any association between the ApoE4 genotype and any protein expression, then all of a sudden, if you select the top 250, you generate this like pretty clear split in the PCA. And I think it's important to recognize that that happens whatever the source, whatever the there's no biological content to which proteins I change their expression of, right? So that clustering doesn't actually tell you anything about the underlying biological signal, except that the patients that had more ApoE4 genes had some amount of changes in protein expression across this giant data set. It could have been because the patients that had more ApoE4, just by chance, were from a different socioeconomic status than those that didn't have ApoE4. There could be other underlying population dynamics. There could be other underlying age dynamics. Whatever other associations are coming along for the ride that weren't measured or weren't tested are going to drive this split. And it is not necessarily meaningful. So I think that sort of tempers some of our criticism there a little bit. It can't literally be random. But also, I think it is the case that it doesn't provide any additional information. Once you've selected on mutual information, doing the other analyses, certainly the machine learning, certainly the PCA, once you've selected your top mutual information, doing those other analyses doesn't actually add any explanatory power. It doesn't do anything new.
And
over just selecting the top from the list.
Yeah, and we're sort of pulling this magically out of the air. And prior to jumping on with this, you walked me through the code and explained a little carefully or more carefully.
And I'm going to post.
Yeah,
I'm going to post the code, share it on the website. I'll share the figures I generated. So if you have computational expertise, you want to go look at the code to see it generated, run it for yourself. We'll make sure that that's possible to do.
Yeah, and the point that I was making is that the amount of mutual information that you need to add in order to generate that split is extraordinarily small.
Yes.
You're talking about, shifting a normal distribution of, 1% potentially of
1% of the genes by a tiny little bit.
By a tiny bit, yeah. Right? And so I guess the point is, will you get this result if you do a full-on random number generation? The answer to that is no. But if you deflect for any reason into that biological system to where really any mutual information is contained, you do. It's sort of an expected outcome that these PCAs would look the way. So you can rule out that there's no information contained within this giant proteomic screen in this huge set of patients, and that's good. But we have to be really careful as to, because I don't know, when I look at PCAs, right, and you see movement like that, we tend to think in magnitude, right?
Yeah.
Like it takes a lot of movement in order to drive those things when you're doing like a transcriptomics based analysis, right? And I think the point you're making is that because we are selecting on mutual information specifically, our eyes are going to detect a change in the PCA clustering that is much less significant than we are used to looking at when we're looking at PCAs.
This just occurred to me. So the amount of noise that I added, I mean, I can't say this definitively, but like, I imagine if I collected a bunch of samples in Boston and processed them, and Matt collected a bunch of samples in Emory and processed them, there's going to be some batch effect. And if by chance the population in Emory had more ApoE4 than the population in Boston, the batch effect from the difference in collection and processing would be sufficient that if I then did mutual information feature selection and a PCA, then I would see that split.
Yeah, so we're on a hair trigger. I guess that's the point. Exactly. Is we're on a hair trigger. Like if these are perfect samples and there's no difference in underlying biology, then what you're seeing is, potentially some amount of mutual information shared. But we're, like I said, we're on a hair trigger, and there could be some pretty significant confounding factors here. I guess that's the point.
Yeah. All right. So that's one piece of the analysis. So the other piece of the analysis that we have spent or will spend discussing is the enrichment analysis. And we sort of, when we recorded yesterday, we sort of talked about some of the potential problems here. And so I went and I pulled up the PantherDB tool. It's on the web. You can go find it too. And again, I've got code to generate lists. I will put that on the website alongside this episode. Basically, what it comes down to is we are asking about enrichment of a set of genes over baseline. And what this tool allows you to do is upload a list of genes. You can have numerical values as well, but you can also just upload a list of genes that are selected and ask, is that enriched relative to some other set? And so the right way to do this, excuse me, is to upload a list of your enriched genes. So in their case, the ones that they selected based on mutual information, and then also upload the reference list. So if they took the list of SomaScan proteins and included it, I tried a bunch of different ways to generate these lists of proteins in a sort of random way. And if you upload the correct reference list, then no significant results are returned by this tool. So if they did that, and it's fairly easy to do, then you would expect no significant results if there is legitimately no biological signal. It is unclear from the methods if that's what they did. Another option on the website is to select as your reference list human genes. And if you do that, even with a random subset of the 7,000 proteins from SomaScan, you get a bunch of significant hits. If your reference is the whole human genome. And right. I want to be very clear that we are not saying that that's what this group did. It is very easy to do that. And one of the things I think that's worth highlighting, not as a critique of this paper necessarily, but it's just as a warning for anyone else doing this analysis, that you need to think very hard about what the analysis is. And if you sort of blindly click around on a website like PantherDB, you might end up generating results that are not legitimate. And again, I want to be 100% clear, we are not saying that that's what this paper did. I can't tell based on the methods, but it is possible that that's what they did. It is possible that if you're trying to do this analysis, it might be what you do, and so you just want to be careful that you're not generating.
And the reason that this happens, right? And this is increasingly something to think about. The reason this happens is because these are commercial products that are being produced, right? So SomaScan is a commercial product. It's an aptamer-based, and we don't have to talk about exactly how it works, but it's an aptamer-based proteomics assessment tool, right? They have 7,000 proteins. When you build that commercial product, the question that you're asking yourself as you're validating one gene, one protein after another, after another, after another, is what are the 7,000 most useful proteins that biologists are going to want so that they purchase this scan tool, right? Now, when you're asking yourself that question, you're not asking yourself, what are the first 7,000 random proteins that I could offer? They are, what are the 7,000 proteins that people are most interested in? And exactly. Frequently, when you're talking about soluble proteins, you're talking about things that are immune-related, cancer-related, cardiovascular health-related, right? All of those things. And you can even generally buy those kinds of subsets from these companies, right? If you have a cardiovascular health question, you can buy a 2,000 protein set, for example, that'll target those things, which is great. Good tool, right? Except that if you then take that and you ask the question, are my biologic samples enriched against the human genome? What you're going to get is a bunch of cardiovascular health hits. And the reason for that is because you've already selected 2,000 proteins associated with cardiovascular health, right? Of course exactly. They're enriched. You're not measuring the rest of them, right? And so, I think that that's the concern here, is that these commercial platforms are frequently skewed towards immunology for good reason. People buy them for exactly that reason. They did here. And if we are not careful and we don't recognize that that is an underlying enrichment, there's a real concern that you could accidentally run these things the wrong way. Now, like Kevin said, no indication as to whether that's happened here, but we have seen that happen in the past with these kinds of things. And the challenge here is that without that being explicitly called, this is the fundamental observation of the paper, right? The title of the paper is ApoE4 carriers share immune-related proteomic changes across neurodegenerative diseases. The only place that there is any indication of inflammatory effect is through these GSCA analyses, right? And these things are not mutually exclusive, potentially, right? So if you have that underlying noise that skews you mostly towards inflammation, oncology, all of those things, and then a handful of genes that may or may not be real, probably are, but a handful of genes find their way, proteins, I keep saying genes, proteins find their way into these proteomic sets, then you're going to sort of use the noise as a platform to then highlight something that probably wouldn't have come up significantly, but is now significant because that noise isn't being appropriately accounted for. So again, do not know that is what we are looking at here. Do not know. Could well be that this analysis was done correctly and that, the inflammation signatures here being correctly reported, although I would say that, it would be nice on reflection if we had some smoking guns here. Like if IL-6 was up and they tested IL-6, if TNF alpha was up, if some rock solid component of the T cell receptor signaling pathway, B cell receptor signaling pathway, if TLR7 is up, if those sorts of things are in these data sets, it would be great to see those things because they would make the paper, I think, much stronger. Even simply pulling out some of those sort of rock star immune components and showing a simple bar plot would have been really helpful here because it's hard to come away with those specifics. But maybe it's there. Maybe they are reporting that signature correctly, but it's such a critical component of this paper and the final findings that I think it is absolutely worth making sure that we're crystal clear on how this analysis was done.
Yep, all agreed. Okay, so with that aside, now we're going to go back to the main episode somewhere. We don't know yet where.
Yeah. What? So, with that in mind, so we, I don't know where to take it from there, Kevin, because here's the problem with that is, so we've found 229 genes that seem to correlate at some level with ApoE4 homozygosity, right? We throw them into a GSEA. It seems to be that inflammation is going to be the thing that shows up most frequently. There is some concern, it sounds like, at least from your interpretation of what in reality is really being told to us through these various pathway analyses. I can tell you that, we move on to immune cell enrichment assessments, which is a not new, but new-ish way of, taking a data set and basically asking the question, we don't actually know what immune components are here, but based on these proteins that we see, what are the most likely associated immune subsets? We do this in spatial transcriptomics just a little bit. It's sort of, just trying to take genomic or genetic information or mRNA expression information, sort of infer what our cellular compositions might be based on how they're distributed and the kinds of. It's fine. It kind of works. My worry here, though, is that we've really trimmed back the number of proteins that we could associate with these things, right? So, it's hard for me to understand how confident we can be using this 229 protein set in making fine discrimination, for example, in the difference between a memory B cell and a naive B cell, right? Or a gamma delta T cell versus an NK cell, right? Like I believe that they're getting real results here, and that it's telling them something about the potential for biology. But my experience with these sorts of algorithms is that there is a little more flex than you want there to be. And so, that doesn't mean, by the way, that this is wrong. What it means is me, the immunologist, I think, in reviewing this paper, is looking at this and thinking to myself, show me that you have a bunch of naive T cells in the CSF of these patients then. Before I believe that, I would like to see trafficking of naive T cells in an unimpaired ApoE4 patient.
I don't think that's what's happening here, though, right?
Yeah.
What they're doing is they're saying these proteins are associated with these cell types. They're not actually saying that these cell types are in the CSF.
Right. I assume... Right? Yeah, I mean, that's true. My assumption would be then that...
So again, if the top hit was glucose 6-phosphatase, just involved in glycolysis, every cell has it. If the gene that you found was glucose 6-phosphatase, sorry, would that show up in this list as a T cell gene? I kind of think it would. Right?
Yeah.
That's and also, looking at these heat maps, these do not look substantially different from random noise. These heat maps. Am I wrong?
I don't know. I don't know. I don't really know how to interpret this.
I feel really bad because part of me is convinced this paper being in Nature Medicine means that I must be missing something. I feel I want to be clear that it is entirely possible that I am missing something fundamental about how these analyses were done, and that all of my criticism might just be based on some misunderstanding. I don't think that's the case. I would say it if I thought that was the case, but it could be. And 3 reviewers, presumably, thought that did not come up with the same criticism.
Yeah, that we know of. Although I do think, did this get to open review? It'd be interesting, actually, to go on in this one on reviewers' comments if they publish the reviewers' comments. Okay, well, yeah, with all those questions, I think that figure 2 becomes very easy to talk about, because basically what happens here is we move from the CSF of these patients to the blood of these patients. We find, basically we do similar things. We take the 6,000 plus proteins from the blood analysis. And now we've got a couple different disease designations. There's some ALS patients in here, frontal temporal dementia in here. And we run that PCA and once again find no real way to pull out correlated signatures associated with these different things. And then they go in and they calculate mutual information again on these proteins. And now we have 58. So substantially fewer genes now that are associated, that ApoE4 status clearly gives you information about protein expression. They do functionally what we did in figure 1, which is to run another PCA. They found that once again, the correlated genes that they identified as a part of the larger set does indeed help you stratify whether the underlying genetic predisposition, which you sort of selected on in choosing those genes, that does get you a split in the PCA, or at least a biasing in the PCA. Perhaps not quite so much of a split, but also now you only have 58 as opposed to your 229. They run the same heat map. We run the machine learning analysis, and then we go back into GSEA analysis and find functionally the same thing a second time. There is no clear difference between the gene signatures or the protein signatures that show up here versus in the CSF. We've got viral processes as the number one FDR. Apoptotic processes are in there. And then on the other side, EBV infection, Hep B, Hep C, and then some TNF signaling pathways, things like this. So, I mean, functionally, a mirror image, right, of what we saw in the CSF. And I think the argument here is that these are shared processes, that the ApoE status is telling you something about inflammatory state, not only in the CSF, but also in the blood in a way that you can identify. Is that a fair read of what's being argued here?
Yes.
Okay.
Yes. So if you want to be the most generous, and I mean, honestly, I do want to be the most generous. I really do.
It's a good goal for us all.
Yeah. What you would say is these are big data sets. We already talked about these are hard to generate. Like just collecting all the samples, real tough. And they identify the associated proteins with ApoE, allelic variants, in a group of non-impaired controls. And they show that the proteins that are associated in non-impaired controls with ApoE are shared in folks with disease and seem to be enriched for immune-related pathways, according to this analysis. And it holds between CSF and blood, the pathways that are identified. So there's some independent, like setting aside all of the like PCA and setting aside the machine learning, which I think just sort of recapitulate the fact that these things have mutual information. We're enriching for particular pathways in those associated genes. In two different body sites, that is like some level of independent confirmation. The challenge, as I said, is the databases that we have of protein function that go into these pathway analyses are not themselves unbiased, right? That we do not know the function of all genes. The way that they get annotated into pathways is based on how they were discovered and how they were identified and how they've been researched in the past. So if a particular protein, let's say, is involved in a viral process and is also involved in lipid metabolism, which is completely, like, would not be surprising necessarily that a protein would be involved in multiple biological processes. If no one's discovered the lipid metabolizing process, it's going to show up as a viral process and not lipid metabolism. That doesn't mean that it shouldn't be in lipid metabolism, right? So, like, the thing that I am concerned about with the pathway enrichment analysis is that for whatever reason, and the fact that the which ones filter to the top is so correlated between CSF and blood actually raises this concern. Like, you might say, well, it's independent confirmation, but it actually increases my concern that it's a feature of the underlying pathway annotations, not a feature of the alleles themselves. And when it comes down to it, like, the thing we actually care about is not which variant of ApoE these folks have, it's the disease that they have. And they're not showing associations with disease that are separate from the associations with the gene.
Yeah, and in fact, they really emphasize that in Figure 3, right? And I wanted to ask you, because this was something that I have not encountered before. I have never seen a stacked bar plot of combined FDRs for a pathway. And that's not to say, by the way, that is not a legitimate thing to plot. I have just not seen it. And what they're showing is that basically across all of these disease states, you're getting functionally exactly the same Epstein-Barr related protein enrichment, for example, and hepatitis B and viral carcinogenesis, right? So what they're showing is essentially that, or they're trying to show, I think, is that there's actually very little correlation with disease state here. ApoE4 in general drives an environmental inflammatory state present because of the genotype, not because of the disease itself. I think that is what's being argued here, and I just have not seen a stacked bar plot of FDRs before, and I looked at that and I was like, "Am I reading that?" Like,
it makes zero sense here. It makes. Okay. High horse on data visualization. When you are doing data visualization, there are certain implications to certain kinds of plots that our brains just naturally go to. And you want, as far as possible, to align the sort of like default assumptions our brains make with the type of visualization that you're doing. In the case of a stacked bar plot, typically that would be if you want to show like proportions of a whole that are attributable to different categories, or you would want to show that like the combined weight, the quantity of something is more in one thing than in another thing. The log FDR of enrichment is not a quantity that it makes sense to compare like this. It just isn't. It's not like a number of genes. It's not. It's not a quantity. It doesn't make sense to plot it as combined quantities. And the comparison is just weird. Like, if you want to show for the different diseases how they all stack up, it just no, it doesn't make sense. But again, like the take home here, mm-hmm. like the heat map in B and D, which I think shows the same information, is a much clearer representation of the data.
Yeah.
Right?
I assume that it is the same data replotted.
It seems that way. When I just look, incidentally, they flip the y-axis. I'm guessing because whatever they're plotting library did, like the higher values go higher in the bar plot, but they're lower on the the heat map, right? So it's like these are sort of nitpicky details, but it like, to me, a lot of these things we're talking about smack of let's throw everything at the wall and see what sticks. And that's not a way to do. That's just not a way to do this.
Yeah. Okay. So, I mean, potentially being generous, we have this really nice data set. We've done an enormous amount of work to generate proteomic signatures in the CSF and in the blood. Those signatures have not been as robust as we wanted them to be necessarily. And so we maybe are digging a little bit deep to try to find pathways that are associated with these signatures that we have, to be very fair, worked our asses off and spent a lot of money on. And so I think what we are looking at here is, sort of the downstream of that. Does seem certainly, again, to associate more by genotype than disease state, although certainly, we do see some movement in ALS, for example, maybe in the intensity of some of these pathways, although, again, we're not clear exactly what we are necessarily looking at. And so then we get to figure 4, which I am worried about talking about a little bit, because I honestly, I do not understand this figure, because it seems to me like the implication to the lower part of this figure is that these genes can only correlate with one thing. But I don't know if I'm reading that right? Like it seems to me like if you try to understand an intersection and a correlation between age and FOXO1, that because of the way that you've colored this, there's only one disease that could be associated with. And I don't understand why that would be the case when we're seeing so much overlap between these pathway analyses, right? So FOXO1 and age. Is it true that it only associates in Parkinson's disease? There's no association, and it's binary here, and I don't quite get it. But maybe I am missing something here that.
No, I missed that implication entirely, but I think you're right. I'm just going to reread that section real quick. Because that does seem bonkers. Okay. Our analyses revealed unique neurodegenerative disease-specific significant relationships with ApoE. That's figure 4b. And that's just it. This analysis further identified that ApoE epsilon 4 central node proteins are significantly correlated with demographic, clinical, and lifestyle variables in a disease-specific way in ApoE E4 carriers.
So, to me, that argues, so they test. They've got, 20 genes here or so? Let's say 15, 20 genes?
Yeah.
And they're testing specific correlation relationships between those and features of these individual patients. Age, congestive heart failure, all of those things. And they're saying that in that matrix, which must have, 150, 200 correlation tests at least, they find no individual correlations between a gene and a risk factor that is shared by any two disease states. Is that right?
My guess, so the description in panel in 4C is categorical heat map showing the unique neurodegenerative disease-specific relationships.
Right.
So my guess is they saw a bunch of other correlations that were not disease-specific. Mm-hmm. This heat map, or this panel, they call it a categorical heat map. That's not a heat map. It's just a... this is not something that I would normally nitpick, but it demonstrates some fundamental sloppiness in my brain. And that might be ungenerous.
There's no heat. There's no heat. Yeah.
There's no heat. This table is showing the associations that were disease specific.
Right.
Not all of the other ones. So all of those white boxes could have been correlated with two or more diseases. And these are just the ones that were specific. That's my guess.
Okay. Interesting.
Which again...
Yeah, so my brain said no correlation.
Right.
But you're right. The the probably correct interpretation was complete correlation or correlation with more than one disease. So,
because frankly, I'm like,
okay,
yeah,
that could be. That could be.
It's not a way to display data.
I don't know that I have much more to say about the data in this paper. To be honest with you, like I think that we have extracted probably most of what we can extract here. Figure 5 is a summary of everything that we just said. I mean, they based on everything that you've heard in this discussion so far, I think probably an average listener would walk away thinking that we're probably questioning just how robust the immune phenotypes are presented in the paper and defended by the data. Figure 5 pretty confidently asserts those immunologic associations in a way that probably, if you've been listening for this last hour and a half, maybe you think that we are wondering about and would like to see a little bit more validation for. And to be fair, I think here we are in the early phases of this field. This strikes me as a paper that tries to go in an unbiased way and really create or cast a very exploratory net along a rich patient data set to try to give the field directions. And I suspect that is, why it is in Nature Medicine. I guess the question that we always have coming to the end of this is like, where are the gaps here? I think, we've probably highlighted a lot of those gaps along the way. For me, from a cellular immunology perspective, I think, the obvious question that comes from all of this is, if this is a measurable peripheral blood inflammatory state, that comes from somewhere, right? This has cellular implications. It would be, I think, really important to know if we have measurable differences in immune composition, if those differences in immune composition reflect functional differences, if we see differences in T cell responses, B cell responses in these patients, how do they respond to vaccines? There are plenty of other downstream implications, I think, that this would have. And of course, that's before you get into the potential for actual engraftment of cells into the CSF, into the brain, right? All of those sorts of things, I think, which are sort of the long-term downstream implications of what's being argued here. Do you have, if we believe the antiviral response stuff, do you have the cellular signature of an antiviral response in the blood? Would we expect to see the cells circulating naturally in these patients that we see during like a mild autoimmune sort of phenotype or a vaccinated, whatever that must look like? But I feel like at the very least, this paper leaves open, I think, a lot of exploration in the immune space and how that immune space really impacts what we are seeing with progressive neurodegeneration across the board. Is that fair?
I think that's fair, and I think it is, for all the snark, this paper is a huge amount of work, and unfortunately, these authors could not have published a Nature Medicine paper saying, we did a shit ton of work to get tons of CSF and blood, measure all of these proteins, and we don't see a lot of strong association. They just couldn't do that, right? Because of the way that our publishing system works. Even if that were true, it may be that their conclusions are correct. And frankly, like, it wouldn't be surprising if given what we know about the biology, it would not be surprising if ApoE variants are connected with immune system processes, and that those immune system processes are related to disease. It could be very hard to identify that. It could be that you need many more patients to get statistical significance, or it could be that their analysis is correct and we are just wrong for some fundamental reason. But regardless, this is a big amount of work, and it is worth giving them credit for that. I would not feel confident bringing any of these results into the lab to start to do experiments on. Maybe that's the bottom line. Like, this maybe generates a bunch of hypotheses and needs other ways to validate. And maybe all of this snark is unfair, but it feels like there's some fundamental lack of what is there a lack of? All of the words that I first reached for don't feel fair, but I just looked. Not just back when we had first mentioned it, I looked at who there are 2 reviewers that are named and 1 anonymous reviewer. Mm-hmm. The 2 reviewers that are named are both experts in fields of neurodegeneration. Couldn't quite tell. I think one of them might be a physician. But they're both aging neurodegeneration researchers. They're not like statisticians or machine learning people, which makes sense for reviewing this paper. On the other hand, the foundation of every result in this paper is a quantitative thing, and I may be concerned that wasn't quite understood in the review. I wasn't able to find the actual reviews themselves to see whether these concerns were raised, but it's. I feel bad shitting on this paper, but
and yet you have.
And
here we are. No,
yeah.
No, I mean, it is. I think part of. It's part of the scientific process, right? It's part of learning how to do science, to dig into a paper and to decide what it is we are going to hang our hat on and what we will not. And, part of that is self-preservation. Because, like you said, if we're going to build on this, you have to be confident that what you're seeing is true. And I think that the conclusion here is we would probably want to see more than this before really digging in and spending a lot of money to try to replicate and maybe you could, right? Like, maybe you could go back to this data set and, do a targeted inflammation panel and, do something like that. It does not escape me here that despite all the immune implications, we don't walk away with a gene signature that's heavily focused on immunology here. Like you said, Kevin, I think a lot of the genes certainly do show up in immune cells, but it doesn't feel to me like the B cell receptor, for example, right? Antibodies, we're not walking away feeling like, there is a clearly defined set of obviously immune-driven response genes. What we have here is sort of a haze of sort of associated inflammatory markers without the real smoking guns, right? Like if you saw IL-6, right? Like if they said IL-6 is statistically significantly upregulated in ApoE4 drivers in the brain or carriers in the brain, right? That would be an immediate interesting thing, I think, to a set of immunologists, right? Because that has a ton of implications. I don't know that we walk away from this paper with that kind of thing, which it just makes it hard just makes it hard to know where to fall.
Maybe another way to think about that is like if you were imagining taking these results and saying like obviously the end result of a lot of this research is we want to be able to identify therapies, right? Like for folks that are homozygous for this variant, we want to be thinking like, are there early interventions? Are there things we can monitor? Like you were saying earlier, like are there particular biomarkers we can look at to see, like, is it progressing slower or faster in these folks? Are there remediations that we can do to try to interrupt, whether it's inflammatory processes or metabolic processes or vascular processes? And we have a lot of immune modifying drugs, right? Small molecule drugs, antibodies. So for autoimmune disorders, we've got all kinds of things that we treat people with. And so if you were going to try to take these results and say, like, it would be unfair for me to say, like, what's the therapy that would come from this paper? Because it's much earlier stage than that. But, like, what's the direction that you would drive in? Like, if you were going to say, like, okay, I want to now I want to do some in vitro studies to look at the results, the effects of some gene on neurons or on microglia or on T cells or on B cells or whatever. Like, what would you do based on this? What hypotheses are you generating based on this that you would be confident taking in vitro or into an animal model? And like, none of the data as presented here, like, gives me any direction.
Fair enough. Well,
yeah.
That's Kevin has learned nothing.
I've learned that correlated things are correlated. That seems important. I have learned a tautology. That's mean. It's mean. I'm sorry.
All right. Well, this has been Audiomunity, episode 50.
Episode 50, Matt. We got to episode 50.
We did.
That's exciting.
We're really limping through, I would say. What's the 10 years or so? So
yeah, but let's say like 3 active years.
Yeah, I would say
4 active years.
Three active years. So yeah, maybe the denominator is a little different. No, 50 episodes. We have periodically shifted the number of folks that are on here. I think that that will continue to fluctuate here and there. I think we'll just keep it going. We'll see if we get to 100. We'll see how that all works.
I think we can. I think we can get to 100. I'm wondering how to put this on my CV, and I was wondering, like, should we post these episodes to PubPeer as like post-publication peer review?
Yeah.
Or this is scholarly output of a kind.
It is. There's a real risk that everyone just hates us if we do that, though.
Certainly in the field of neurodegeneration, people might hate us.
Yeah.
That said, most of the time we don't savage people. Last episode we had nothing but praise for Nicole Ackermans and her review paper on headbutting. Mm-hmm. I think we're going to have more guests in the future. We've got a couple in the queue. If you like what we do here, or if you want to email us and complain, please send us an email, contact@audiomunity.org. Audiomunity is spelled AUDIOMMUNITY.org. At our website, audiomunity.org, you can also find all of our past episodes, RSS feeds, links to our YouTube channel where we're posting these videos. If you could rate us and review us on any podcatching service that you use, I think iTunes is the most important. We have a small, dedicated audience. We would like to grow it. We'd like more people to hear our snark and our compliments. Some episodes are more heavy in one than the other. But please, the most important thing you can do is share us with your friends. If you are interested in coming on as a guest, if you or a trainee of yours in anything even moderately associated with the immune system, as we said last time, you should send us an email, again, contact@audioimmunity.org. If you want to support us, you can go to patreon.com/audioimmunity, and throw us $1 or $2 an episode. We really appreciate it. The music at the beginning and the end was composed by Rachel Reineck. Anything else we want to say? We should do episode 51 as like a retrospective and prospective on audioimmunity. But I think that's it for now.
All right.
Sorry for the snark this time. Yeah. I think I am going to splice in that video. All right. See you next time.
Sometimes you get lucky.